Okay, so we have seen that which steps involved in the biosynthesis of cholesterol. So in that we have seen that this first one is mevalonate synthesis from the acetyl coenzyme A. Then from mevalonate we can synthesize isoprenoid units. Isoprenoid we can say we can contain six isoprenoid units to form squalene, and squalene on cyclization gives lamosterol, and finally we got cholesterol from the limestone. So we will see now step one by one all steps. So here is the schematic representation of all steps I have given over here. So here you can see at the starting we have started from the acetyl coenzyme A. Okay. Then from acetyl coenzyme A we do get something in between. Then we got mevalomic acid over here. Okay. So all this is biosynthesis, and that one this all this cholesterol synthesis occurs in animal body or human body. Okay. So this is this and different enzymes they do catalyze this reaction. Okay. So here we got mevalonic acid. Once we got mevalonic acid, from mevalonic acid we can go for mevalonate 5 phosphate. Okay, so mevalonate 5 phosphate. So here you can see mevalonate kinase. All these kinase and all these carboxylase, all these are enzymes, and they do catalyze all these reactions. So then after that one, we from mevalonic acid and from all these different derivatives of mevalonate, we can get isopentenyl 5 pyrophosphate, that is IPP unit. Okay, so here we got IPP unit, and this IPP unit again we are going to convert to the geranyl pyrophosphate. Okay, so geranyl pyrophosphate again we can add one farnesyl pyrophosphate. Okay, farnesyl pyrophosphate we can get farnesyl pyrophosphate. Okay, so once we got farnesyl pyrophosphate, then we can convert farnesyl pyrophosphate into squalene C30 carbon atom 330. And once we got squalene, we can convert squalene to 2,3 oxido squalene, and once we got oxido squalene we can convert this squalene in lanosterol. Okay, so once lanosterol we got, we can go for the cholesterol. Okay, so this is the schematic presentation of how we can go step by step from the acetyl coenzyme or from the mevalonic acid or from the mevalonate to the cholesterol biosynthesis. Okay, so these are the steps. Now we will see one by one all these steps how to do the synthesis of the cholesterol. So first one is the stage first. The first stage in the synthesis of cholesterol is the formation of isopentenyl pyrophosphate from acetyl coenzyme A. Okay, this set of reaction which takes place in cytosol starts with the formation of 3-hydroxy 3-methyl coenzyme A from acetyl coenzyme A. Initially, two molecules of acetyl coenzyme A condenses to form acetoacetyl coenzyme A catalyzed by cytosolic thiolase. Okay, so we have to synthesize acetoacetyl coenzyme from the acetyl coenzyme A. Okay, so acetoacetyl coenzyme A condenses with the further molecule of acetyl coenzyme A catalyzed by whatever enzyme to form HMG coenzyme A, and this is reduced to the mevalonate by NADPH. Okay, so in this way we can get the mevalonate. So what we have done, two molecules of acetyl coenzyme A. We have taken two molecules of acetyl coenzyme A, and we did condense these two molecules of acetyl coenzyme A to the acetoacetyl coenzyme A. Okay, so two moles of acetyl coenzyme A condensed together to form acetoacetyl coenzyme A, and this acetoacetyl coenzyme A condenses to further molecule of acetyl coenzyme A. Okay, so now 1 mole of acetyl coenzyme A and 1 mole of acetyl coenzyme A. These 2 moles it gives the mevalonate. Okay, and this mevalonate is catalyzed by NADPH. So in this way, we got here our first step, mevalonate synthesis. So here is the schematic representation of how to synthesize the mevalonate. Okay, mevalonate here I have given. So everything I have written over here. So step first is biosynthesis of mevalonate from the 2-acetyl coenzyme. So here is the structure of 2-acetyl coenzyme. Okay, So from 2-acetyl coenzyme A, we can go for acetoacetyl coenzyme A. Okay. So here I have given explanation for all this thing. Once we got acetyl coenzyme A, we can again add one more acetyl coenzyme A. Okay. So pay attention on the number of carbon atoms. Okay. Number of carbon atoms they do increase. So here first three two carbon atoms. Now here one, two, four carbon atoms now. Now here one, two, three, four, five, six, seven carbon atoms. Okay, so like that, go on counting the carbon atoms. Now in mevalonate, one, two, three, four, five, six carbon atoms and two hydrogen groups are there in mevalonate. Okay, so this is the biosynthesis of mevalonate from 2-acetyl coenzyme A. Okay, so now we can go for how the mevalonate we can convert for the in the direction to synthesize the cholesterol. Okay, so step second. Now we formation of isoprenol units from the mevalonate by loss of carbon dioxide. So we are going to synthesize isoprenol units. Okay, so isoprenol units, as we know, it has five carbon atoms. So production of isoprenol units in a three-step reaction catalyzed by kinase, mevalonate is converted to 5-pyrophosphomevalonate, which on decarbonization forms isopentenyl pyrophosphate (IPP). Okay, so it is isomerizes to dimethyl allyl pyrophosphate. Okay, so IPP and DPP they are interrelated. They are isomer to each other. Okay, so first we will synthesize isopentenyl pyrophosphate (IPP) unit, and this IPP unit on isomerization gives DPP dimethyl array pyrophosphate. IPP and DPP are 5 carbon isoprenoid unit. Okay, so this is the DPP and IPP is 5 carbon isoprenoid unit. Then that one we have synthesized from the mevalonate ester. Okay, and mevalonate ester we have synthesized from the acetyl coenzyme A. Okay, so once we got this 5 carbon unit isoprenoid unit, we can combine all these isoprenoid units. We can go for synthesis of squalene. This is easy, not that of difficult. So here is the how to synthesize isoprenoid unit. Okay, IPP, isopentenyl pyrophosphate. Here at the end we have synthesized. Okay, so how do we have synthesized? So we have taken here now my mevalonate. Okay, mevalonate we have taken here. So mevalonate converted into 3-isopentenyl pyrophosphate in three consecutive reactions requiring ATP. So I think the ATP is catalyzing the reaction. Okay, so pay attention at how mevalonate is converted into the 3-isopentenyl pyrophosphate. So first we have taken mevalonate on treatment with ATP. It gives out ADP. So phospho groups is getting attached to the hydroxyl. Okay, H from the hydroxyl group from the alcohol group. Has been replaced by the phospho group OPO4. Okay, sorry PO4. Again on treatment with the ATP, next molecule of ATP, another molecule of phosphorus is getting attached. Okay, so now again more treatment with the ATP, another phosphate group has been attached, phosphorus containing group has been attached to this hydroxyl group now. At the end, elimination of carbon dioxide. Here this carbon dioxide is getting eliminated. Okay, so to form this kind of unit, so carbon carbon double bond we have got over here, and pyrophosphate. Okay, two phosphorus atom with one bridge oxygen atom. This type of isoprenoid unit has been formed here, and we are going to use this isoprenoid unit for the structure or for the synthesis of the cholesterol. Okay, so once we got IPP, okay, so isoprenoid unit we got, we can convert this IPP to GPP. Okay, geranyl pyrophosphate, and from GPP we will go for the farnesyl pyrophosphate. Okay, so these types of all these sequences for conversion on how to do this conversion we have seen already in our terpenoids also. Okay, Uh, so pay attention on that one. So isopreneur units, they are very important for the point of view for synthesis of these terpenoids, and the same sequence we have taken for the synthesis of cholesterol. Okay, so we'll see now one by one how to convert the this now IPP 3 IPP to the uh, geranyl pyrophosphate or farnesyl pyrophosphate. Okay, so we'll see now. So now here mevalonate, they started from the mevalonate. Okay, mevalonate, they have started mevalonate kinase. We got mevalonate 5 phosphate, again treatment with ATP. Mevalonate 5 diphosphate, this one we have converted here isopentenyl diphosphate. Okay, so here we got isopentenyl diphosphate. Okay, so this isopentenyl diphosphate 3,3-dimethyl, we have to isomerize that one. IPP is isomer of DPP. Okay, so this isopentenyl diphosphate we have we this one is in this one we have converted to the 3,3-dimethyl allyl diphosphate. Okay, so remember, if exams will be online, then you have to draw all these things uh, in your answer sheet. So be careful regarding the structures. Okay, count the carbon atoms, carbon numbers, and then you can draw the structures. Okay, so once you got DPP here, dimethyl allyl diphosphate. Okay, DPP you got here from the IPP, isopentenyl diphosphate, converted to the dimethyl allyl diphosphate. Okay, so from DPP we are going to generate the geranyl pyro diphosphate. Okay, so geranyl diphosphate we got over here. So count the number of carbon atoms in DPP: 1, 2, 3, 4, 5. Okay, so 5 carbon atoms in DPP. Again here isopentenyl diphosphate also 5 carbon atoms: 1, 2, 3, 4, 5. Okay, so number of okay, position of this carbon carbon double bond is different only in these two structure, DPP and IPP. Okay, now next we will go for the geranyl diphosphate GDP. Okay, so now we count the carbon atoms: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That means now 2 moles of DPP or 1 mole of DPP, 1 mole of IPP we can combine together to get this geranyl pyrophosphate having carbon number 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So two isoprenar unit, they do combine with each other in presence of different enzymes. We can get the GPP. Okay, so geranyl diphosphate. Now geranyl diphosphate. Okay, geranyl diphosphate, we can convert geranyl diphosphate to the farnesyl diphosphate. Okay, so now I think in farnesyl diphosphate definitely there will be the 15 carbon atoms: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, so 3 in geranyl pyrophosphate, geranyl diphosphate, sorry, geranyl diphosphate, we again geranyl diphosphate we can add one isopentenyl diphosphate. Okay, IPP unit to get this farnesyl diphosphate. Okay, so not that of difficult on this cholesterol mechanism. Just go step by step, count the carbon atoms, and then you can go for the writing the different structures of IPP, DPP, and IPP. They to convert into the farnesyl diphosphate. Okay, so now we'll see now in next slide how to use this farnesyl